Hello, biology students. You will be happy to know that we are getting into a topic that is a lot more familiar to a lot of people and a little bit easier to understand than cellular respiration and photosynthesis. We are now getting into mitosis and meiosis and the cell life cycle, and this is something that people have at least heard of before and are a little more comfortable with and is a little easier to follow the process. So we'll be talking about the cell cycle, eukaryotic chromosomes, mitosis and cytokinesis, cell cycle and how that relates to cancer, and then prokaryotic cell division. So the cell cycle is a set of orderly events that allows the cell to make a copy of itself. It starts off as one cell and then it divides into two, and those two can later divide into four. We see this happening more often with an embryo that is developing because those organisms have to divide very rapidly. For adults or grown organisms, we don't see this happening quite as often, but it still occurs within every living organism. So before the cell is ready to divide, the cell grows larger, it doubles the number of organelles, so it will have two Golgi bodies, to eat smoothie R's, things like that. And the DNA also gets replicated, which we'll talk about in a different chapter. DNA replication is actually pretty straightforward and not too difficult either. The two major stages of the cell cycle is first interphase and then the mitotic or division stage, which includes mitosis and then cytokinesis. So this is a really great picture that you can come back to and refer back to. It shows how most of the cell's life cycle takes place in interphase. So we have G1, S, and G2, which are the growth phases, and then we have mitosis, and mitosis is actually a very small part of the cell life cycle, but this is when division is actively occurring. Most of the cell life cycle takes place in interphase. So interphase is when the cell is doing its cell thing and just being a cell and being normal without any real complications. If it's a skin cell, it's being a skin cell. If it is a stomach cell, it's being a stomach cell. That's what this phase is. And the cell, it varies how much time is spent in interphase depending on what that cell is. Some of them need to go through mitosis faster than others. Nerve cells and muscle cells do not complete the cell cycle, and they remain in the G0 stage. So interphase consists of G1, S, and G2 stages. It takes 90% of the cell cycle, so even though it's often overlooked and we focus most of the time on prophase, metaphase, anaphase, telophase, which are the mitosis phases, this is really a very vital part because it does take up the vast majority of the cell. So G1 is the cell is in recovery from the previous division, and it's just kind of increasing the size of the organelles it's got. It's going to grow in size. It's also going to accumulate materials that it needs for DNA synthesis. The S phase is the DNA synthesis phase. During this phase, it's going to replicate its DNA, and it's also going to make sure that it's got enough materials and start preparing for the cell division that's coming up. But mostly the S stage stands for synthesis stage, so that the cell is making copies of its genetic material. The sister chromatids, will see that they are the result of what is attached when a cell replicates itself. And then the G2 stage is located between DNA replication and the onset of mitosis, and this is the cells making proteins. It has microtubules that develop, and it's necessary for the division. Just kind of double checking and making sure, sorry cat, and making sure that the correct materials are present for the next mitosis stage. So here you can see a chromosome. Now what's important to understand is this is one chromosome with one set of DNA. When we see this, we call it a sister chromatid. And with a sister chromatid, the materials have been duplicated. So we have one set of chromosomes on this side. This is one set of material, and this is exactly the same material. So it's duplicated itself. We've got one set, the same thing, genetically the same. Something that's important in mitosis is that everything gets replicated the same, because we don't want any mistakes. Mistakes are bad. We want a stomach cell to stay a stomach cell. We want the skin cell to stay a skin cell. We don't want any mistakes to occur here. So making sure this process replicates exactly is super important. They remain joined at this centromere that you can see here that joins the two halves of the chromosome. Then we enter into mitosis. Now, mitosis is the division of the nucleus. This is different from cytokinesis. Cytokinesis is kind of a different set, but the cytokinesis is the division of the cytoplasm only. Mitosis is the division of the nuclear information. So mitosis is division of nuclear and DNA. Cytokinesis is division of the cytoplasm. The point of this is to result in two genetically identical daughter cells. Again, we want them to be the same. Mitosis, we want everything to stay the same. So a signal is an agent that influences the activities of a cell. The cell cycle is controlled by external signals, such as it grew too big. If it's too big, the cell can't manage it, and so it needs to cut itself in half and go through mitosis in order to reduce its size. There are family of proteins called cyclins that increase and decrease the cells as the cell cycle continues, and specific cyclins must be present for the cell to proceed from one stage to the next. This is important because it's the way the cell can check itself. If a cell is malfunctioning, you don't want it to make a copy of itself. You only want it to make a copy of itself if it's running efficiently and if the cell is okay. Without cyclins, the cell stops at the G1, M, or G2 phase, and that's what we want. We want the cell to stop itself from reproducing if it's not a healthy cell doing what it's supposed to. So damage by solar radiation or X-rays is repaired at the G2 checkpoint. So if we have a damaged cell, we can fix it up to a degree, as long as it's not too severely damaged. Now, if the cell is severely damaged, it will go through something called apoptosis. Apoptosis is programmed cell death. So the cell can kill itself and destroy itself and reabsorb by other cells if there's something wrong with that cell. And this is good because we don't want a damaged cell going and making other damaged copies. This is how things like cancer can happen. If a damaged cell gets out of control and keeps reproducing more damaged cells, that's one of the main causes that causes cancer. So apoptosis is an important part of getting rid of these bad things. Apoptosis is caused by enzymes called caspases. And cell division and apoptosis are kind of opposing forces. But again, it's going to be something that's necessary if the cell's malfunctioning. So here we can see normal cells, and the cell rounds up and the nucleus collapses. We can see that eventually the chromatins condense in the nuclear fragments, and these little things are called bleaves. That's kind of a fun word, like extra divisions. Cell fragments contain DNA fragments, and we can see eventually the cell starts to disintegrate, an apoptotic cell, and it's starting to get broken down and will get reabsorbed by other parts of the body. Cell division and apoptosis occur throughout our lifetime to maintain homeostasis. There's that word again. Abnormal cells that could become cancerous or virus-infected cells are cells that die through apoptosis. Cells harbor this enzyme that are normally kept in check by inhibitors, but it's there if we need it. And there are signaling proteins that will say, hey, something's not going right. We're not going to make a copy. Let's pause here and maybe even destroy the cell if we can't fix it. So there are regulations in place to say, hey, this is good, we can go on, or hey, it's not good, let's pause this and maybe if we can't fix it, destroy the cell. And so these checkpoints are vital in order for the cells to reproduce healthy. So the eukaryotic chromosome contains a single DNA molecule in a double helix. The DNA is associated with something called histones. Histones are a type of protein that help to wrap up the long DNA strand so that it can be transported correctly. So histones play a structural role and have good essential survival factors that help it to organize the DNA overall. Nucleosomes are wrapped up around, it's a DNA wrapped around a histone, and it kind of looks like beads on a chain, and it helps again just to organize that long DNA strand. So here we can kind of see how it works. We've got our chromosome. Okay, humans have 46 chromosomes, 23 from each parent. This chromosome has already been duplicated into a sister chromatid. So we've got one chromosome, and here's its exact copy. So we got our DNA strand that's in a double helix, and it wraps around a little histone here. The histone is the purpley kind of protein area, and it usually loops twice around a histone. Then we've got several of these histones that are joined together. Eventually, that's going to get organized in a neat structure and wrap it up. Now, the reason that this is important, if you were going to a store, like a craft store, to buy a bunch of yarn, if the yarn was all unraveled, can you imagine the mess that would be to try and take all this unraveled yarn into your basket and up to the register, put it in a sack, you've got yarn spilling out everywhere. Well, for moving DNA, it's the same thing. The DNA strand is very, very long, and it would be straggly and hard to maneuver. But if you wrap that yarn up in a nice, neat bundle, it's super easy to throw it in your cart and go check out and then bring it home. That's what the chromosomes are doing with these histones and these nucleosomes. It's taking something that's long and scraggly and putting it into a nice, neatly wrapped bundle so that it's really easy to transport when the cell is getting ready to divide. Now, a more highly compacted form of the chromosome is called heterochromatin, and this is an inactive chromatin, and genes are hardly ever described. Now, we haven't talked about transcription yet, or genes and how they fall on chromosomes. We'll get into that later. But most chromosomes have both compaction levels, some that are really compacted and some that are not as compacted. So mitosis and cytokinesis. Eukaryotic cell division involves mitosis and cytokinesis. Mitosis is nuclear division. Cytokinesis is the division of the cytoplasm. During mitosis, the sister chromatids are separated and distributed into daughter cells, so that X shape is going to be pulled apart so that each side of the new cell is going to have the exact same information. So before mitosis even begins, the chromatin is going to condense and become the chromosomes, and it's going to make a copy of itself in the S phase of interphase. And each species has a characteristic chromosome number. We already mentioned this, that it's 46 for humans, 23 from mom, 23 from dad, for a full set of 46 chromosomes. Here are some examples of how this can vary. 46 is what we have, but it's really crazy what some of these other organisms can have. Yeast has 32 chromosomes, a garden pea has 14, a potato 48. This adder's tongue fern has 1,320 chromosomes. That's a lot. Fly has 8, humans 46, and a goldfish has 94. You'll notice that all of these are even numbers because they get half from each parent. Diploid means the number includes 2 sets of chromosomes of each type, and that's what we are. We are diploid. We have half from mom, half from dad, and diploid can also be represented as 2N. So each of the cells in our body, with the exception of reproductive cells, are diploid cells. So humans have 23 different chromosome types, and the eggs and sperms are the only ones that are actually haploid or 1N. Haploid or 1N is half of the information, so think haploid half, and that's 23 chromosomes. And the point of that, again, is an egg has 23, a sperm has 23, they combine together, 46 for a full set. So during interphase, a cell prepares for cell division, and organelles are duplicated, including the centrosomes. And at the end of the S stage, each chromosome has internally duplicated, and we have the sister chromatids that are joined at the centromere. During mitosis, the centromeres will separate, and they will get pulled to opposite cells so that we have the exact combination. Here's what a chromosome looks like again. Here's our centromere. Now the kinetochore, that's a little different. The kinetochore is important for, we'll look at spindle fibers in a little bit, and those spindle fibers are going to join to this kinetochore. So one chromosome and its genetically identical sister chromatid, and we've got sister chromatids joined together here at the centromere. All right, so just outside the nucleus is the centrosome, and this is going to build tubules for the cell, and these tubules are going to help orient the cell and move the different chromosomes around. In animals, the centrosomes contain two barrel-shaped centrioles, and we've talked about the centrioles before. The centrioles are going to form spindle fibers through them. These spindle fibers are going to connect to things to move things around. They also assist with lengthening the cell, getting ready for that cytokinesis portion. So in prophase is the first step of mitosis. So one way you can remember it is IPMAT-C, I for interphase, P for prophase, M metaphase, A anaphase, T for telophase, and C for cytokinesis. The PMAT, prophase, metaphase, anaphase, telophase, those are the mitosis steps. Interphase and cytokinesis are not part of nuclear division, but they are part of the overarching cell cycle. So chromatin is condensed in prophase. Chromosomes can now be seen if you're looking at it under the microscope. The nucleolus disappears and the nuclear membrane starts to go away. So the nucleus is starting to dissolve because you don't need the nucleus when you're getting ready to divide. Spindle fibers are beginning to form, and the two centrosomes begin to move away from each other. In animal cells, these microtubules are going to form star-like arrays that are called asters, and this is eventually going to help the cell kind of elongate in kind of like an oval shape. Sometimes you can divide the stages out, and that's another important thing to remember is even though we learn this as prophase, metaphase, anaphase, telophase, it's a smooth continuous process. So if we did a lab with this class, we would look at these under a microscope and we would see that there are different in-between stages, and that's why you might see something like prometaphase. It's not quite prophase, it's not quite metaphase, it's kind of that in-between, which is completely normal since it is a smooth continuous process. But this is preparation for the sister chromatids to separate. The centromeres and chromosomes develop the kinetochores. They are specialized proteins complex which allows for connection with those microtubules. So the microtubules are going to connect at the kinetochore. Metaphase. Metaphase, think middle. Metaphase, middle. Metaphase is basically the chromosomes lining up in the middle. That's really all they do is line up in the middle. And they are positioned there based on those spindle fibers. It's called a metaphase plate, and there are some overlapping of the spindle fibers because that's going to help the cell elongate too. There's an M checkpoint that delays the start of anaphase until the kinetochores are attached properly because we want the sister chromatids to separate right. We don't want one cell to end up with too many chromosomes and another cell to not end up with enough. During anaphase, think apart. Metaphase, middle, anaphase, apart. Or you could think of it as away because the sister chromatids are being pulled apart at this process, at this stage. So now we're having cells pulling those chromosomes that were joined and they're no longer joined together. Telophase, think 2 because we have 2 new cells that are forming. The nuclear membrane is going to come back, the spindle fibers begin to disappear, and the cells start to decondense, the chromatids start to decondense and start to go back into their spindly DNA form. The reason that the DNA does not stay as a chromosome or compacted and wrapped up in a nice neat bundle is because it is not able to make proteins and perform its other functions if it's all wrapped up. It needs to be stringy for that to happen. So it only really gets wrapped up during the mitosis process. There are now 2 sets of everything we need, and cytokinesis will typically start during this part. So again, cytokinesis is not the same thing as mitosis. It's not a part or step of mitosis. Cytokinesis is a separate step, and basically it just happens at the same time as telophase. Sorry, throat tickle. So here we can see the process taking place in both animal and plant cells. On the top we have mitosis for animals, on the bottom we have mitosis for plants. The stages will be a little bit different for cytokinesis with plants because we do have a cell wall to worry about in plant cells. So here we can see the typical cell in interphase, early prophase. It's starting to condense, but we don't have a really condensed membrane yet, but you're starting to be able to see some of those chromosomes be apparent. Here we have prophase that's full on. You can see these asters that are forming and starting to join those spindles to the chromosomes here in the middle. Nuclear membrane also starting to go away. Then we've got the prometaphase, so more joining, that nuclear membrane mostly gone. It's starting to stretch out, but it hasn't lined up in the middle yet. So metaphase, middle, and there is a centriole, and here's another centriole, and the fibers, you can even see the tiny little fibers joining them. Here is another metaphase plate, metaphase for middle. Then anaphase, you can definitely see that they are pulled apart from each other. They are no longer lined up nice and neat in the center. The chromosomes are being separated and pulled to either side. In telophase, we have a distinct pinching in of the cell. That's actually cytokinesis taking place. And we can see that they're starting to unwind. We're starting to have the nuclear membrane come back. And we see a cell plate form in between the cells for plants because plants have that cell wall. So here are just the steps in kind of a good comparison, but taking them step by step. We're going to go through these kind of quick because we've already talked about them, but paying attention to these microscope ones especially, you can see how it's different for plants versus animals. Here we can see the actual prophase, and then we've got our prometaphase, and then our metaphase, which is taking it and lining it up at the plate. Anaphase, they're being pulled apart. Telophase, also called telophase. I just always call it telophase. And cytokinesis is also happening here. Now, cytokinesis is different for animals or plants, which I've already mentioned. Cytokinesis is the division of the cytoplasm. And this helps make sure that each cell has the correct amount of cytoplasm necessary. And usually it just pinches off right in the middle. And that's the cool thing about animal cells, is because there is no cell wall, and we've already talked about the fluid mosaic model, that these animal cells can just pinch off in the center. They create something that's called a cleavage furrow, so think of it as like a rubber band around the middle, and it pinches in and pinches in until eventually it pops off into 2 different cells. This cleavage furrow is going to appear between the daughter nuclei and is formed by a contractile ring, and it pulls in like a drawstring, so like a rubber band, making it smaller in the center. Eventually it'll pop into 2, and some gets cytoplasm from the mother, and some gets the original cell, and the other also gets cytoplasm from the original cell. Now with plants, this is different. In plants, we have the cell plate that forms because there's a cell wall. We can't pinch in like we did with the animal cell. With plants, the cell plate starts in the middle and works its way outwards. And so it's going to form that original, it's going to have like vesicles line up in the center. And these vesicles have little pieces that can be made into a cell wall. And as these vesicles fuse with each other, the cell wall components are going to fuse, and a cell wall is going to be made in the center. So with the plant cells, it grows from the center outwards. The cell plate grows from the center outwards. And with the animal cell, it's going to be pinched in from the outside in. All right, so here we can see cytokinesis in an animal cell. The pinching in and the cleavage furrow, and eventually it's going to be pinched in all the way to the center until the two cells kind of pop off. These cells look kind of hairy because remember they have a lot of proteins on the surface. Here we can see the plant cell forming. It's a series of vesicles. Those vesicles have the materials necessary to make a new cell wall. They're going to join together in the center, and that is going to create that cell plate. So the reason that we have mitosis is it growth and repair. So the only way a multicellular creature can grow is if they have more cells. And these cells, it's not that we have bigger cells, it's just that we have more cells. And so you will see a lot of mitosis in an organism that is quickly growing. That's why in a lab you would do a mitosis slide from the root tip, typically of an onion, and the root tip is constantly growing. Or a whitefish blastula is another common one that you're going to see in the lab. The whitefish blastula, a blastula is a fish that is still in the egg that's developing. So something that is growing really fast is going to have more mitosis taking place. Embryo becomes a fetus. After birth, the child becomes an adult. The other thing that the other time you might see mitosis taking place is if there's an injury. If you get a paper cut, you got to fill in that hole, and it's going to be filled in by mitosis. The cells that are next to it are going to start going through mitosis to fill in that gap. So to grow or to repair something that's damaged. Two reasons mitosis may happen. Now, there's also something called stem cells. Stem cells are an area of controversy, and many mammalian organs contain them. They're specifically found in organisms that are developing, though, like think of little babies. They have more stem cells. And stem cells are basically cells that can become any other kind of cells. They are not programmed to have a purpose yet. And that's why it's so interesting to do research on stem cells. Because if a cell doesn't have a set purpose, it could become any other kind of cell. The stem cell could become a stomach cell, or it could become a heart cell, or it becomes a liver cell. It doesn't have a purpose yet. If a person had a heart attack and you put stem cells near their heart, these stem cells could potentially turn into heart cells and repair the heart naturally. So you can see why this is an amazing area of research that doctors are very focused on. Therapeutic cloning is an example of how stem cells can be researched, or how stem cells can be used and are currently being researched. And this is the idea to produce human tissues, and can begin with either adult stem cells or embryonic stem cells. And say a person needed a heart transplant and there were no hearts available. Could it be possible to grow a heart by using stem cells? And if it had the right nutrients and also, of course, a blood supply, could we do that? Could we grow an extra organ by using stem cells? And it's a high area of research because the transplant list is so small and it's so hard to get an organ if you need one, and then to find a match. If you had a heart that was genetically the same as your own, there'd be a highly less chance of there being complications with that surgery. Embryonic stem cells can be used for reproductive cloning, the production of a new individual, and there has been successful cloning in the past, although it is still a very high area of controversy. So two types of cloning, the therapeutic cloning and the reproductive cloning. So in this case, you have an egg and you remove the nucleus. So now you just have like this ball, and you can put a new nucleus in. So you take that egg and you take a nucleus of something you want, like this is how Dolly the sheep was formed. They had an egg from a donor, and then they had a cell that was taken from an adult sheep, and they took the nucleus out. They put it in that egg cell, and then they basically zapped it with some electricity to get it functioning. And then they implanted it into the surrogate mother, and eventually it did make a clone because the genetic nucleus that they put in had the exact same DNA as the one that they cloned it from. Now, therapeutic cloning, kind of the same idea, only in this case they can culture out those embryonic stem cells and use that to determine what they want. They can say, okay, we started off with the same thing, didn't really have a purpose, but we can make it a nerve cell, a blood cell, or a muscle cell, depending on what is needed at that time. So the cell cycle in cancer, if cells grow abnormally and they continue to grow in a way that we don't want and our body is not catching it, then this can lead to cancer. Now, sometimes those tumors are benign. If you have a tumor and you hear benign, that's good. That's what you want to hear. Benign means that it's not cancerous. It's sometimes in a capsule, and that capsule has prevented it from spreading, and it doesn't typically invade neighboring tissue, so it stays in one place. Benign tumors are typically pretty easily removed, and the chances of them growing back are very, very, very slim. You do not want to hear them say malignant, though, if you have a tumor of some kind and they say it's malignant. And a malignant tumor is cancerous, but it is not encapsulated. And that means that it is invading area tissues. A lot of times what happens when something is malignant is it can also do something called metastasize. Metastasize is super dangerous. What happens with metastasize is some cancerous tissues and cells break off into the bloodstream or the lymphatic system, and it travels to other parts of the body. And that means that you may have had liver cancer, but all of a sudden you've also got pancreatic cancer, or you might have a brain tumor forming because it has metastasized and it has traveled to a different part of the body and started a cancer there. So metastasize is the most dangerous. But anytime you have malignant means that it is a cancerous cell that is dividing. There's usually no clear-cut edges, which makes it very difficult to remove, because think about mitosis. If you have one damaged cell that is left, that damaged cell can reproduce and make more and more damaged cells. That's why some people will go into remission and then their cancers come back, because there was a hidden cancerous cell somewhere that just took a little time to start reproducing to become noticeable again. Development of cancer tends to be gradual and multi-steps and may take years before cell is obviously cancerous. So sometimes it can take a really long time before the cancer develops. So things that cause cancer cells is cells are non-specialized. Cells are immortal and can enter the cell cycle repeatedly. They just divide and divide and divide, and that's why cancer can progress very quickly. They could have an abnormal nuclei where it's enlarged, an abnormal number of chromosomes, too many or too few, and they don't make copies the way that they're supposed to, or something gets separated out wrong. A lot of times cancer cells are also not going through apoptosis, or that cell death. So instead of dying like they're supposed to when there's something wrong with the cell, they're making copies of themselves over and over again, which is not safe. Cancer cells are abnormal but fail to undergo that apoptosis process. Form tumors, which is another issue. Mitosis is normally controlled by the contact of the neighboring cells, and contacting and feeling a cell that's next to it stops the process. But with cancer, it doesn't. It just continues to grow and grow and grow and goes out of control. The metastasize, again, this is super dangerous. The fragments get into the bloodstream and travels to other parts of the body. It'll come out somewhere else and start a cancer there. Undergo angiogenesis, and they form new blood vessels, and they bring nutrients and oxygen to the tumor. So they're actually bringing stuff to that area, because a tumor needs growth and it needs oxygen and blood to supply it. And this is why people who have really large tumors are losing a lot of their nutrients, because a lot of their nutrients are being diverted to that tumor to feed it. So here we can see the primary tumor and a new mutation or something that occurs, a lymphatic vessel and a blood vessel. Right at this point, it's not super concerning yet. It's possible to even take out surrounding tissue, and a lot of times surgeons will do that just to make sure they get it all. They will take out surrounding tissue just to be sure. However, if it's left untreated, it could go into those lymphatic vessels or the blood vessels, and this is when it's going to metastasize. Metastasizing means it's going to come out somewhere else and build a new tumor in that area. Now, cancerous cells are non-differentiated. They don't really have a purpose, although there are some types of cysts that are kind of weird, and the cysts will grow like teeth or hair in random parts of the body, and that's kind of strange. You could have a cyst on a female ovary, and it has like little teeth or fingernails in it. So sometimes the cells can be differentiated but the good thing about those weird kinds of cysts, those typically do not have they're not cancerous. So even if you have a tumor that's growing like teeth in your stomach, good sign is it's not cancerous, at least. So non-differentiated cells for cancer, abnormal nuclei, do not undergo cell death, no contact inhibition, they're just going to keep growing, disorganized and metastasized. Normal cells are differentiated, normal nuclei, they do undergo cell death if something is wrong. If they touch another cell, they realize, hey, I should probably stop growing. Organized and remain in the original tissue and don't go into other tissues. So the origins of cancer, they think it could be because of external factors such as skin cancer, for instance, or exposure to radiation on a regular basis. Or it could be a genetic factor, a genetic mutation. So ONOC genes are proto-ONOC genes, code for proteins which promote the cell cycle in various ways. And a proto-ONOC gene is mutated, it becomes an ONOC gene. There's tumor suppression genes that try to help our body not produce tumors. And they code for proteins which inhibit the cell cycle and promote apoptosis in various ways. And sometimes these can be damaged and not doing their jobs properly and not catching when a cell is damaged and just allowing it to grow continuously. So we can see that sometimes there are receptors that should tell the cell to die, but they don't. And sometimes it's not catching when a cell is having issues and allowing the cell to reproduce when it shouldn't. So proto-ONOC genes are normal genes which are part of a stimulatory pathway. They promote progression through the cell cycle and they recruit receptors and signals. Mutations in these proto-ONOC genes cause them to become ONOC genes. And this can stop a cell from dying or stopping mitosis when something is wrong. And it just lets it continue with the process even when something is wrong. The tumor suppressor genes can become inactive, and instead of suppressing tumors, it doesn't do that, and it allows the cells to continue to divide. And some tumor suppressors promote apoptosis, and if these are not working correctly, then the cell is not dying the way it's supposed to. A mutation in a tumor suppressor causes a cell cycle to accelerate, and now it's dividing quick and quick and quick, and that's again why sometimes cancers can progress at a very high rate. Half of human cancers involved an abnormal or deleted p53 gene. The p53 gene turns on the expression of other cell inhibitory genes. So this p53 gene, if you've got that, it tells the cell, okay, let's make sure that we're doing things right, and if this is broken, then it can cause a lot of other things to break too, like a chain reaction. Alright, so chromosomes normally have special material at each end called telomeres. And telomeres, these get shorter after each cell division. When they get very short, the cell will no longer divide. This is one of the theories for why people age and die, is because at some point your cells are too tired or too old to divide again. Because remember, according to the cell theory, cells came from one original cell, that first embryo, and then all they've done is divide since then to make you as you are. And eventually all that dividing is going to wear them out because the telomeres get shorter. Telomerase is an enzyme that maintains the length of the telomeres, and if telomerase gets mutated, then the telomeres will shorten, and then that causes the cells to die. So it allows cancer cells to continuously divide over and over and over again. Alright, so last but not least, the prokaryotic cell division. It's a little bit different because you don't have a nucleus to work with. So prokaryotic bacteria and archaeobacteria, chromosome is in a ring, and DNA and a few associated proteins are assisting with this. Folded up in an area called a nucleoid, so it doesn't have a nucleus, but it has kind of a area where it's found. And 100 times the length of the cell, or sorry, 1,000 times the length of the cell. So it's really long. Okay, there's a lot that's coiled up in there. It will be replicated into 2 rings prior to cell division, and the replicated rings attached to the plasma membrane, hopefully, on different sides of the cell. This allows for binary fission, which is splitting in 2, and the 2 replicate chromosomes are distributed to the 2 daughter cells. So one ring gets in one daughter cell, the other ring gets in another. They are identical, and this is how a lot of single-celled creatures are going to reproduce, is they just make a copy of themselves. The advantage is if it wants to make a copy of itself, then that's good, and it doesn't need another organism to reproduce. However, the disadvantage of this is if there's something wrong with that one cell, yeah, you just kind of made your offspring also have something wrong with it. So E. coli is an intestinal microbe, has a generation time of about 20 minutes. 20 minutes. It can make a copy of itself and have a new generation. So this is why E. coli can spread very quickly on materials that are like infected with it. So comparing and contrasting prokaryotic and eukaryotic, binary fission and mitosis both ensure that the daughter cells are genetically identical. Prokaryotic and protists are single-celled, and cell division of single-celled organisms produces 2 new organisms, which means basically they just reproduced by cell division. In multicellular, more complicated ones, the cell division is mostly for growth and for renewal or repair if something is damaged. Binary fission, the chromosomes duplicate and each daughter cells get 1 copy, and there are no spindle fibers involved in binary fission because it's joining that the plasma membrane instead. So here's just looking at binary fission. We've got our ring of DNA and the nucleoid, and then that is going to make a copy of itself. You see a red and a purple one here, and then it just basically splits down the middle, separating out those copies from each other until you have 2 completely new cells that are genetically identical to the original. So these have resulted in the same, and here's our genetically identical one. So again, just to compare and contrast, the prokaryotics, for cell division, they have binary fission, and that's asexual reproduction is what they're going to do. In eukaryotic, they could go through mitosis and cytokinesis, and sometimes it's for asexual reproduction, but in more multicelled creatures, it's for growth and repair, and if something is damaged. All right, yet another chapter down. We are flying through these. If you have any questions, let me know, and I will see you in class. Bye, guys.